Here we are in AP Biology Unit 6, Gene Expression Regulation. This is video 2. This video is going to cover DNA replication, which is Unit 6.2, as well as mutations. We're jumping ahead and we're going to cover mutations, which is 6.7. All right, let's start with DNA replication. When and why is DNA replicated? DNA is replicated during the S phase of interphase, and that prepares for both mitosis and meiosis division. So what process exactly is it that adds new nucleotides to a DNA strand during replication? So how do we get a single nucleotide added to a growing strand of nucleotides or nucleic acid? And then also, what provides the energy for this process? Well, the name of the process is dehydration synthesis. That's the general process for adding monomers to a growing chain to create a polymer. The energy here comes from the release of two phosphate groups from that free nucleotide. So the free nucleotide has 3 phosphates, and during the process of adding to the growing chain of nucleic acids, it loses 2 of those phosphates to provide energy for the process. So how does this process relate to the direction of DNA formation? Take a look at this, and you notice we've got our strands labeled 5' and 3'. How does the mechanism of adding a new nucleotide relate to this directionality? DNA builds in a 5' to 3' direction, and that's because it's the 5' end of the new nucleotide, where the phosphate is, that connects to the free hydroxyl group on the 3' side of the existing strand. So you can see that in the diagram on the left is that the phosphate, the 5' end of the new nucleotide, grabs onto that OH, that hydroxyl group, on the 3' end of the existing DNA strand. So you always need that free OH group on the 3' end of the growing strand. And that's why it grows from the 5' to the 3' end. Is the 3' end is where the new growth can happen. So we're going in a 5 to 3 direction in terms of building. What enzyme is responsible for this process? What helps catalyze the synthesis of DNA? That's an enzyme known as DNA polymerase. You can maybe remember this is that it's causing the polymerization. We're making a polymer. We're making the nucleic acid. And the polymer that we're making is DNA. So that's why it's DNA polymerase. How does DNA polymerase know which type of nucleotide to add to the strand next? If you remember from the last video, there are multiple different kinds of nucleotides that differ in the base. How does it know which one to add next? So the one it adds next has to do with base pairing rules. And if you see on the right side here, we've got an existing strand of DNA. So an existing strand of DNA is used as a template for the new strand, and new nucleotides are added according to base pairing rules. So what are those base pairing rules? What bases pair with one another? And let's see if we can apply that here. What nucleotide is going to be added to this strand next? So A pairs with T and C pairs with G. So the next one added here is going to need to pair with that open C on the right. So the next nucleotide added is going to need to be a guanine or a G. What 2 factors influence base pairing? Maybe you can look at the diagram and see if you notice anything about sort of the structure of the connection between the bases that might help you answer this question. The 2 factors that influence base pairing are the number of hydrogen bonds, so they need to match. A and T both form 2 hydrogen bonds, so they pair. C and G both form 3 hydrogen bonds, so they pair. But we also need to pay attention to the width of the pair. For example, A could form 2 hydrogen bonds. Why can't A bond with another A? Well, you need to have a 2-ring purine, which is Cu and T, always bonded to a 1-ring pyrimidine, which is A and G. And that keeps the overall width of the double helix consistent throughout, is that we're always going to have 3 rings across, a purine and a pyrimidine. So we said that DNA polymerase is what reads the template strand and what adds those new nucleotides, but how can it read it if all the bases are hidden in the middle of that DNA double helix? This is where we have an unzipping enzyme. So that's called helicase. So an enzyme named helicase unzips the strand. What is it that it's disrupting? What connection are usually connecting the bases that helicase has to break? Helicase disrupts the hydrogen bonds between the bases. And remember that hydrogen bonds are not really a full bond. It's not a covalent bond we're talking about here. The strand on the top and the strand on the bottom are still separate molecules. It's just an attraction between them due to the hydrogen bond. All right, so this unwinding kind of creates a problem. I don't know if you've ever taken a rope and tried to sort of unwind it. And what happens if you do, if somebody's holding the other end, sort of as you unwind, those coils can become super coiled and a lot of tension can build up. If you pull far enough and enough tension builds up, it's going to slow down this process or create so much pressure that it's going to break. You don't want your DNA breaking, so there needs to be another enzyme to fix this process. That's called topoisomerase. And topoisomerase's job is to snip the sort of supercoiled area and let it unwind, let it relax, and then put it back together. So it prevents this tension from building up of a supercoiling due to the unwinding of the helicase. So let's get back to this kind of picture where we see our helicase is starting to unzip or unwind our DNA helix, or separate those two strands. So DNA polymerase can only add nucleotides to an existing nucleic acid strand. Remember, it needs that OH group on the building strand to grab onto. So before DNA polymerase can begin, we have another enzyme involved that creates an RNA primer. So a primer is just something to get something started. What enzyme creates the primer? And I don't know if you've ever heard of it, but if you have, hopefully you can remember from the name of what it's creating. It's creating a primer. So the enzyme that creates the primer is aptly named primase. So primase lays down a little bit of RNA, and that allows our DNA polymerase to get started. So the DNA polymerase can grab on to that primer, that RNA primer, and begin to lay down new nucleotides. Eventually, the RNA primers are removed and replaced with DNA by a different type of DNA polymerase. And then those gaps are sealed up by an enzyme called ligase. We'll return to ligase a little later. So if you look at this diagram, you'll notice that the strand on the top and the strand on the bottom, the new strands, are being built in opposite directions. Why is it that DNA polymerase builds these new strands in opposite directions? This comes back to the function of DNA polymerase, that it needs that hydroxyl group, that OH, to be able to grab onto. So new nucleotides can only be added to the 3' end of the growing strand. And that's because RNA polymerase needs that hydroxyl group on the 3' end of the growing strand. Because the parental strands of DNA, those are the ones that already were existing, those are the old strands, because the parental strands are anti-parallel, you'll notice that the one on the top is running 3 to 5, and the one on the bottom is running 5 to 3, if we're reading left-right. So those are running in opposite directions, which means growth of the new strand is also going to occur in opposite directions. But something that's consistent, whether we're talking about left to right or right to left, DNA is always being read from 3' to 5'. So the old DNA is always being examined by DNA polymerase in a 3 to 5 direction. And the DNA polymerase is adding new nucleotides in a 5 to 3 direction. So we read from 3 to 5 and we build from 5 to 3. On this diagram, you might notice the terms lagging and leading. So the top is, in this case, the lagging strand, and the bottom is the leading strand. What does that mean? The leading strand builds in the direction of the replication fork, so it can just keep going as more DNA is unwound by the helicase, whereas the lagging strand builds away from the replication fork. So if you look at the top, that's the lagging strand. That's pointing away from the replication fork. And the replication fork is where helicase is acting. And then that one on the bottom, the leading strand, is heading in the same direction as helicase. So as helicase unzips more DNA, that leading strand is going to be able to, DNA polymerase on the leading strand is going to be able to just follow helicase and keep building and building and building. So how does this difference in direction lead to a difference in the types of strands that are produced on the leading and lagging strands? So to answer this, let's take a look at a little mini animation of replication in action. So we can see that this double helix is about to be unzipped by helicase, And as it gets unzipped, we start the process. That green is showing the primer, and then the red is showing the new DNA. So we begin this process. So on the top, we have what's the lagging strand, because it's going away from the replication fork. And on the bottom, we have the leading strand, and it's building in that, they're both building in a 5 to 3 direction. But you notice on the bottom, the leading strand is building towards that replication fork. So as we continue, the one on the bottom can keep going, whereas the one on the top kind of runs out of room. And so it needs to start over with a new primer and create another short strand. And as it unzips more, same thing happens. The lagging strand needs to create another little bit, and the leading strand can just keep going because it's unzipping in the same direction as the movement. So on the top, we see that the lagging strand produces short chunks of DNA called Okazaki fragments. And on the bottom, the leading strand produces one long continuous strand of DNA. So those chunks on the top, those Okazaki fragments, we don't want our DNA in chunks, so we need to have a way to connect those fragments. Think back to an enzyme that helped kind of glue things together. The Okazaki fragments of the lagging strand are connected by the enzyme ligase, and ligase is that connector enzyme. So here's a chance for review because we just went over a whole lot of terms, a lot of enzymes. So take a look at this diagram and identify as many terms as you can in this diagram. So some of the important terms that you'll see here are helicase, primase, primer, DNA polymerase, 3' end, 5' end, parental or template strand, leading strand, lagging strand, Okazaki fragment, and ligase. See if you can find all of those on this diagram and see if they make sense, and if not, maybe back this video up a little bit. All right, another important concept here, or rather than just terms, let's focus on the concept. DNA is read in the 3 to 5 direction and built in the 5 to 3 direction. That means there's going to be a difference in how it's built on these two strands. Building takes place smoothly on the leading strand. In this case, we're seeing the leading strand on the top of this diagram. And it creates Okazaki fragments on the lagging strand. In this case, we're seeing the lagging strand on the bottom of this diagram. All right, another little bit to put this in context. What is this yellow diagram showing us? The yellow diagram here is showing that DNA replication takes place bidirectionally, or it's taking place in two directions at once. So we have DNA replication beginning at the origin of replication, and then its helicase is, there are actually two helicases active going in opposite directions. So we have a replication fork on the left, and that's what's blown up in a sort of big version in most of this diagram, but we also have a replication fork over on the right, and both of these are happening at the same time. All right, final concept in terms of DNA replication. DNA replication is known as semi-conservative. What does that mean? See if you can figure it out from this diagram. Semi-conservative means that every new DNA double helix consists of one strand of old DNA bound to one strand of newly synthesized DNA. And you can see the old DNA here pictured in blue, and that's what's being unzipped, unwound by the helicase, and then the newly synthesized DNA, synthesized by that DNA polymerase, is shown in red. All right, so sometimes things don't go always perfectly. And so now we're into Unit 6.7, where we're going to talk about mutations. So DNA replication is amazingly precise, but it's not perfect, and so we can get mutations. What is a mutation? Mutation is a change in the nucleotide sequence of an organism's DNA or in the DNA or RNA of a virus. What can cause these mutations? So mutations can occur naturally during DNA replication, and they can also be due to environmental mutagens, and those include UV light, X-rays, and various chemicals that can cause DNA damage. So what happens if we end up with a mutation? What are some different possible outcomes of a mutation? Well, one possible outcome is that it's caught right away. Sometimes mutations happen as part of the replication process, and DNA polymerase is constantly proofreading and catches itself right away. It tries out a nucleotide, nope, that's not the right one, throws it out, gets a new one. So sometimes those errors are proofread and corrected immediately. Sometimes they're not caught quite so fast, and that the error is only detected after the DNA is synthesized, but it's still pretty new. It then can be repaired if it's still a relatively new segment by a section being cut out and then new replacement DNA be put in, and that gap is sealed. Let's do a review. What's the name of the enzyme that synthesizes the replacement DNA? Think about this as creating new DNA. And then what is the name of the enzyme that seals that gap between the new and the old DNA? So the enzyme that creates that new DNA is DNA polymerase, and the enzyme that seals that gap is ligase. A note on this diagram, that second one there where we can see that gap, DNA can be cut in either the middle or the end of the strand. If it's cut in the middle, the enzyme is called an endonuclease, and if it's cut at the end, it's called an exonuclease. What kind of enzyme do you think would be active here, looking at this diagram? Would this be an endonuclease or an exonuclease that would have made this cut? This would have been made by an endonuclease because it's in the middle of a strand. So here you can see endonuclease cuts in the middle, exonucleases cut from the ends. All right, so what if our mutation was not caught early on, and it continues to persist in future rounds of DNA replication? What if this mutation sticks with us and continues to be replicated? So what I have here, what I've shown, is the order of bases in that picture up to the left showing those colors. I've just put those in letters, and I've shown the mutation in bold. What's going to happen if this strand of DNA, this double strand of DNA, is replicated with that mutation in there? See if you can figure out what nucleotides will be in the new double helixes. So one of the new double-stranded DNAs will have the mutation and one will not. So the top strand, if that gets copied, that has the mutation. And then that serves as the template, so that mutation is going to be there on both sides of that DNA, or on both strands of the DNA. That bottom one doesn't have the mutation, so when that serves as the template, then the new DNA will not have that mutation. This is an example of what's called a point mutation. What does that mean? A point mutation is a mutation that affects just one or just a very few nucleotides. Here we're seeing examples of point mutation that's just affecting a single nucleotide. What's the name of each type of mutation shown on the right? What kind of change happened, and what do you think that would be called? And then of those, which would be considered a frame shift mutation? The mutation type on the top is a substitution, where one nucleotide is switched out for another. The one in the middle is called an insertion, where we get an addition of a nucleotide. And the one on the bottom is a deletion, where we lose one of the nucleotides of DNA. Insertion and deletion are considered frame shift mutations because they shift the 3 codon reading frame that's important in translation. So what you're seeing in this diagram is, on each of these, DNA, mRNA, and amino acids. So you can see how a change in DNA may affect the eventual order of amino acids. For DNA that does code for a protein, like it looks like this one is, do all changes to the DNA cause changes to the protein structure? And the answer is no, not all changes affect the protein structure. Take a look at this diagram, or this table, and see if you can figure out what these types of point mutations mean. So if there's no mutation, we can see that the DNA is TTC. And then we have different types of substitutions. So if that C becomes a T, it's going to end up coding for the exact same amino acid. That's not going to affect our protein at all, and that's called a silent mutation. A nonsense mutation is if we get if we get a different nucleotide in that codon, that it ends up coding for a stop signal of the protein. That's going to just make a complete mess of things. That's called a nonsense mutation. Missense is where we get a change but it might not be a huge change. So here we get a different amino acid, but the difference of the amino acid sometimes is not a big deal and sometimes it is a big deal. These are called missense mutations, where we get a change in amino acid. There are conservative ones where we get the same type of amino acid. In this case, they're both basic. And on the right, we get a non-conservative missense mutation. In this case, the amino acid it codes for is no longer basic in properties. Now it's polar. And so there's going to be a difference of property, which might make a bigger difference in our eventual protein. So speaking of proteins, let's do a little review. What are the 4 levels of protein folding? 4 levels of protein folding are primary structure, which is the amino acid sequence, secondary structure, which is the hydrogen bonding of the backbone. Third level is tertiary structure, and this is interactions between the R groups. So those different amino acids and their properties are going to come into play. And then the last one, quaternary, is the 4th, and that's where we have different subunits. So different polypeptide chains interacting with one another. All right, another type of mutation other than a point mutation is called a chromosomal mutation. Based on this diagram, see if you can define chromosomal mutation and name each of the types shown below. So a chromosomal mutation is something that affects a big chunk of DNA, a long segment of DNA, sometimes an entire chromosome. Different types include duplication, where you get a chunk duplicated, inversion, a chunk is flipped, deletion, a chunk disappears. Insertion, you can get a chunk of DNA moving from one chromosome over to a different one. And translocation is where it can flip back and forth. You get some DNA from one chromosome switched with DNA from a different chromosome. You can also have chromosomal mutations where you get a whole different number of chromosomes. This can be caused by non-disjunction, like we talked about in a previous unit. And if you have total non-disjunction, you're not going to just have like one extra chromosome. You can have an extra chromosome of all the different types of chromosomes. So if you see in the diagram on the right here, we see haploid and a picture of diploid. Then there's an example of polyploid. There are many types of polyploidy. How many would this be? If haploid is N, diploid is 2N, what would this be? This example of polyploidy would be 6N. There are different kinds of polyploidy. It's relatively rare in animals. There are some animals that do show polyploidy, but it's fairly common in other organisms, including plants. Final question, are mutations always harmful? No, mutations are not always harmful. They can be, mutations can be harmful, but they can also have no effect or even be beneficial. Mutations introduce variety. If there's a change in the genotype that does affect the phenotype, that's going to be subject to natural selection. And in some environments, that may either increase or decrease the fitness. So you just can't tell just by looking at something. You have to also know the environment that the organism is in, what that change has an effect on. All right, we're going to leave it off there for today. This is the end of 6.2 and 6.7.